Hi, everyone, and welcome to this webinar on understanding complex disease biology, where we're going to be focusing in particular on the genetics of long COVID. I'm really excited to welcome two speakers today to talk about this subject. Steve Gardner is the CEO and co-founder of PrecisionLife. It's a really cool company that focuses on gaining deep understanding of the biology that drive complex disease. Today we're going to be speaking about this, particularly in the context of long COVID and how this approach can potentially yield new drug targets and new treatment approaches. We'll also be speaking with Heath O'Brien, who leads on the Sano bioinformatics team and has recently been leading on the genetics of long COVID project at Sano, where he co-authored a research paper on the genome-wide association of long COVID. So two really well-suited speakers to be talking about that topic. We have millions of people that now live with long COVID, so it's a particularly important topic that also provides hope that advances in precision medicine might be able to start playing a role in tackling complex chronic conditions. And so I'm really excited to hear about your perspectives on this specific topic, but also on how what we're seeing in all of that bubbling of activities around long COVID could be applied to other conditions in the long run. In terms of the agenda for today, we're going to talk a little bit about Sano Genetics and PrecisionLife, and in particular, the work that both companies have been doing in the context of long COVID. And then we'll deep dive into a detailed analysis of the Journal of Translational Medicine. We will deep dive on a detailed analysis of Journal of Translational Medicine paper, where we're going to be looking at this precision medicine angle in the context of long COVID. We'll spend a little bit of time discussing what this might mean for pharma companies, but also for patients and for healthcare practitioners. And just a reminder as well that we'll be taking questions in the chat and hosting a Q&A at the end. If the question is not answered today, there is always the possibility to email to ask the question, and we'll do our best to get back to everybody. So I'll start by introducing myself, and then I'll hand over to Stephen Heath. I'm the co-founder and the COO at Sano Genetics. I studied neuroscience and genetics at the University of Cambridge, where I met my two co-founders. I worked in ops at JP Morgan before I studied genetics, and today I lead on business operations, clinical and scientific operations at Sano. In terms of my background, I decided to move from academia to creating Sano because I really wanted to see the impact of the research that I was witnessing and how it could be translated into the real world. And I'll talk a little bit later about how Sano does that. But before, I'd like to hand over to you, Steve. I've obviously introduced at a high level what PrecisionLife does, but I'd love to hear from you in more details what you've been working on and the really exciting things that your team is doing.
Oh, fantastic. Thanks very much, Charlotte. Yeah, I'm also a fantastic believer in this space. I've worked for over 30 years, and I got introduced very early to the Human Genome Project. In fact, Jim Watson was on one of my scientific advisory boards in the early 90s when the ideas for that project were really being formulated. And we've seen that project be both inspirational and frustrating. Inspirational because it's transformed oncology, frustrating because those same benefits haven't come across into more complex chronic diseases. And something as heterogeneous, polygenic, multifactorial as, COVID, long COVID, and more severe forms like ME/CFS are really almost the definition of the most difficult diseases to treat or to get a handle on what is driving the disease biology using existing methods. So that's really what PrecisionLife set out to do. We reimagine the way in which we analyze large-scale disease populations in order to be able to generate those kinds of insights.
Lovely. Thank you, Steve. Heath, I'd love to hear a little bit more as well about what you do at Sano, a bit about your background, and particularly about your experience with the long COVID project.
Yeah, absolutely. I've been on a bit of a journey throughout my career. I started out studying the evolution of symbiosis in lichens, actually, so very blue skies basic research. And I've over the years worked in a number of different areas, plant pathogens, and gradually made the move into human genetics 5 or 6 years ago. And I've been working in academic positions until the last 5 years or so when I moved into industry, trying to have a more direct impact on patients' lives. I've been at Sano for 2 years now, leading on all of the genetic data analysis and particularly the first major project I took on when I joined Sano was this genetic analysis of the long COVID study that we were running at the time. So we, yeah, I guess we'll say a bit more about that in the next section.
Thank you, His. I'll take that opportunity to talk a little bit more about what Sano does in the context of these types of study. Sano is a 360-degree platform for precision medicine studies. It was created with the mission of accelerating the world's transition into precision medicine. As researchers, a crucial challenge that we observed when we were in academia was that precision medicine was moving a lot faster than the current clinical trial infrastructure. And we quickly realized that a major bottleneck in making precision medicine a reality was going to be translating these novel drug targets into actionable treatment for patients. And that meant being able to find the right patients and matching them with the right drug targets for their specific genetic background through clinical trials that, as a result, are much more likely to succeed. So we have built an end-to-end platform that supports precision medicine clinical trials through a separate patient finding protocol where we support the clinical trial recruitment process at every stage, starting with the strategic consultation where we work on defining the gene panels that we should select for a specific condition, the sequencing assays that we need to use to get the most out of the research process, all the way through a patient finding protocol, biomarker testing, and of course, one of the most important pieces in all that, engaging the patient through the research process so that we can get really tangible insights as a result of it. Over a year ago, we launched a sequencing program in the context of long COVID in an attempt to identify whether there was some genetic basis to whether or not someone was developing long COVID or not. And as a result, whether drug targets could potentially be identified, or at least disease mechanisms could be identified in these patients. So I won't go into too much details here because he will be talking a little bit more about that later. And Steve is going to go into a great level of details as to the research paper that looked into all of this. So without further ado, I'll hand over to Steve to talk about PrecisionLife in more details and also about the paper that is the topic of today.
Thanks very much, Charlotte. Yeah, as I said earlier, the addressing the frustrations that the Human Genome Project and the ways in which we analyze data for relatively monogenic disease, seeing that those haven't resulted in the same level of insights and actionable discoveries for complex chronic diseases, we set out to think more deeply about the nature of those diseases and what data it was going to be possible to use. 8 years later, we find ourselves with a platform that can routinely analyze complex chronic diseases. So if you wouldn't mind clicking next slide, please. So in the center here, we have Alzheimer's disease. This is an analysis of early onset Alzheimer's patient population. And it illustrates very nicely what we do. This is what we call mechanistic patient stratification. So in other words, rather than taking the simple clinical observation diagnosis that a patient has, we're getting into understanding the factors that are driving different forms of the disease within different patient subgroups. So the colors here represent different patient subgroups who have a different mechanistic cause underpinning their disease. And so here's a big difference. You know, we're not in that linear mutation causes a change in the gene structure, which causes loss of function. We're talking about diseases that present in many different ways with lots of influences from multiple genes interacting with one another, and then exogenous factors like epidemiology and environmental data. And so to be able to do this level of analysis, you need a platform that is capable of handling multimodal patient data, and also identifying those non-linear interactions between multiple different features. And that's at the heart of why we're different from the kind of genome-wide association studies or other more traditional approaches.
That's fascinating.
Thank you. We've had to get very good at ingesting lots of different types of data along the way, and we have also generated multiple data access agreements, and very happy to count Sano as a partner in these areas. If you click on again, so from an understanding of disease perspective, Alzheimer's is a great example. You know, we've had 130 failed trials chasing amyloid and tau hypotheses, and having new drug molecules going into clinical trials. Those are failing in large part not because the drugs don't work, not because they don't engage the right target, but in large part because you're not selecting the right patients to put into the clinical trial. So what I'm showing on the right are the prevalences of each of those mechanisms in this patient population. And you can see lipoprotein metabolism, which is amyloid and tau, represents less than a third of patients. So if you choose your patients just because they have the right diagnosis, they don't necessarily have the right mechanism. And that's the maximum efficacy you're ever going to be able to demonstrate. And you obviously have a whole range of other processes that are going on that could usefully be the target of drug interventions. So that's what we do. If you click on again, please. We basically use these insights. We develop with pharma companies and Gen AI drug discovery businesses, novel target programs. We use these colored clusters, the sets of SNPs in this case. We use those as patient stratification biomarkers based on mechanism. And that can help us accelerate and de-risk clinical trials. It can help us support the launch of a product by choosing the right patients who are going to benefit from that medicine. And it allows us also to get insights into repurposing drugs. As a business, we've got a lot of those different collaborations. So we've now done over 50 different disease studies. If you click on, please, that's led us to identifying hundreds of novel drug targets and hundreds of repurposing opportunities. Click on again, please. We've started working on the left-hand side here with biopharma companies, with CROs, with the portfolio management teams of pharma companies. And click on again, please. We now have 8 biopharma licensing deals. We have 40 of our drug targets under option with partners. 7 of them have been selected for development. And we have, through our various partnerships, the lifetime potential deal value of about $500 million coming out of these.
Thank you, Steve. That's really interesting. I'm going to hand over to Heath to talk a little bit more about long COVID and the genetics of long COVID, and then we'll spend a bit of time diving into the paper in particular.
Yeah, thanks, Charlotte. I'd like to start by introducing long COVID. In some ways, you would think that long COVID doesn't really need an introduction. Everyone is aware of this phenomenon of people getting an acute COVID infection and then taking a very long time to recover or not recovering at all. after the acute phase of the disease is ended. But I think not everyone really appreciates the cumulative impact that long COVID is having. So over a quarter of people who get COVID have reported ongoing symptoms after the acute phase is ended. And the vast majority of them report that these symptoms continue to impact their day-to-day activities once they've recovered. And over a quarter of people categorize these limitations as significant. So it really has a very profound impact on the people that it affects. In terms of what the symptoms are, fatigue is by far the most common. Almost 3/4 of people report fatigue. Difficulty in concentration, shortness of breath, muscle aches are all reported by about half of people with long COVID. But there are over 200 other symptoms that are less commonly reported. So it's a very, very broad cluster of symptoms that characterize this condition. Sorry, if you go back for just a second. It's most prevalent in females in the 35 to 69-year range, and also in people with disabilities. It also disproportionately impacts people in deprived areas, so it's sort of further exacerbating existing inequalities. And the cumulative effect of this is really huge. Over 145 million Europeans have reported having long COVID symptoms after they've recovered from the acute COVID infections. Next slide. So that is a little bit about the impact that this has on a personal level, which I think most people appreciate, but I think less appreciated is the impact that long COVID is having at a societal level. So we hear a lot about labor shortages and that large number of people haven't re-entered the workforce since the pandemic. That's rarely linked to long COVID, but over 100,000 people in the UK have not re-entered the workplace because they're still affected by long COVID. And this is impacting the UK economy to the tune of £1.5 billion per year. It's a similar story in the United States, where 500,000 people have permanently left the workforce due to long COVID, and this represents about 18% of long COVID patients that still haven't returned to work after a year of their COVID infection. Next slide. Yeah, so I'll briefly mention the traditional genome-wide association approach to studying the genetics of a complex disease like long COVID, because that's the approach that Sano has taken in the work that we've done in this area. So we recruited a large group of people who have a long COVID diagnosis, and also a control group of similar people who were able to rapidly recover from their COVID infection. And we've genotyped both groups and just looked for genetic differences between these two populations. We've collaborated with a large international consortium, labs all over the world, running very similar studies. We've all combined our data into a very large data set, and we've succeeded in identifying one gene that seems to predict the people who were less likely to rapidly recover from a COVID infection. And I think this is a pretty typical experience for a lot of the people doing research in this area. There's been a huge amount of interest, but it's been very slow to really understand the causes of long COVID, and also developing cures that can help people to recover from it. I think there's a lot of reasons for this, but the main one that I'd like to highlight is the long COVID probably isn't a simple single condition. It's really a combination of different conditions with very different clusters of symptoms. So the symptoms are extremely diverse. They also tend to be very non-specific, so they are symptoms that can be caused by lots of other infections and various other causes as well. And we're talking about a chronic condition that people live with for years. Symptoms can come and go that may or may not be related to the long COVID specifically. There's also very inconsistent disease progression. So the overall numbers of people with long COVID are very large, but most people do gradually recover. So the biggest concern is a smaller subset of people who are either recovering very, very slowly or seeing major relapses where their symptoms return quite regularly. So there's a lot of diversity within that, and also diversity in the response to the treatments that are available. Some people can recover rapidly when they are treated, and other people been unable to recover. All this suggests that there's probably multiple biological bases to what we call long COVID, and therefore lots of different genes involved. And if you just pool together everyone that has any symptoms persisting after they've recovered from an acute COVID infection and compare them to people who did rapidly recover, there's too much heterogeneity to really pull apart all of the genetic factors involved. So that's where more sophisticated approaches to combining all this information and identifying clusters of individuals that have a similar biological bases is a very promising approach to try and overcome the enormous impact that COVID's having on society and on individuals.
Thank you, Heath. Steve, I'm going to stop sharing and hand over to you. We are going to spend a little bit more time getting into the details of this research and discussing the really interesting insights that have come out of it.
Thanks very much, Charlotte, and that's a great setup, Heath. I'm going to tell you a little bit about the insights that we have into some of the genetics of long COVID. And this touches on a series of papers that we've written starting in about June of 2020 with COVID-19 itself, before anybody had even imagined that there was going to be a long COVID. And then moving through into MECFS, which is one of the most severe consequences, or one of the diagnoses that follows for many patients who have severe forms of long COVID, and then into the long COVID study itself. So I just want to acknowledge a whole series of partnerships here. Sano was the first data set that we used on the long COVID side. We also have collaborations through Decode ME with many of the ME community. They collect explicitly both MECFS and long COVID patient data, and we're hoping that we'll be able to replicate findings in other data sets. Obviously, the NIH is involved. Many of the studies that I'm going to be talking about will also use data from UK Biobank. And then towards the end, as we think about the future, I'll tell you about some of the exciting work that's going on in terms of where this may lead us for pharma and patients. But going right the way back to May 2020, to Heath's point, it can be quite difficult to segregate pathogen-mediated diseases into clear cohorts. The fact that somebody doesn't have the disease might be because they haven't been exposed to the pathogen, or it may be that they have a particular factor in their immune system, or just some other genetic reason why the virus isn't binding in the same kind of way. So in our very first study, we took mild COVID positive, but mild cases and compared them against severe COVID. And we basically identified a whole series of genes that explained the really unexpected diversity of symptoms. So yes, cytokine storm, the fusion macrophages that we saw in lung tissue in the early days of COVID, into those sort of leaky vasculature microcoagulation effects through endothelial cell function, into neurodegenerative functions and cognitive impairment, well, COVID fog as we called it, and then some of the long-term senescence. So we came starting from a very detailed view of what was actually driving the primary disease itself. We were able to use that to identify a series of compounds that we believed were going to be useful in treating severe disease, and we suggested a bunch of them. This one called utasteride read out and now reduces the severity of disease and the course of infection by almost half. And that's an example of repurposing medicines effectively into the population. And I'll come back and touch on that in the context of long COVID. We moved on from that to ME/CFS, and we did a study that basically took two populations from UK Biobank, independent ME and CFS patient populations. And we identified a series of genes associated, well, SNPs, and then mapped those to genes. Most of the signal that we found, and the same is true in long COVID, were from combinations of 3, 4, or 5 SNPs. So in this case, we found 200 SNPs in total in 84 combinations of between 3 and 5 SNPs. And they have very predictive odds ratios. In other words, the chances of seeing these signatures in patients versus a healthy control population was very high. So they had a decent predictive accuracy. Technically, if there are geneticists on the watching this, none of those SNPs are in linkage disequilibrium. So they're not appearing just because they happen to be close to each other in the genome. In fact, most of them are not in coding regions of genes. So only 9 out of 200 are in the coding regions of genes. And we identified a whole series of genetic associations that map to the sort of cellular processes that we would expect to see. So we see autoimmunity, we see cognitive impairment and neurotransmitter related genes. We see genes associated with metabolism, and particularly the production of energy in mitochondria. We see genes that are to do with sleep disturbance or circadian rhythm showing up as well. So this was in our minds when we started some of the additional work, and particularly looking at the overlap, because, as I said, many patients who have long COVID have progressed into an ME/CFS diagnosis. But this shares that same heterogeneity that Heath was talking about. Now, we want to deconvolute that. So these are those same 200 SNPs. What we have done is identified subgroups. So this is bringing a mechanistically defined subgroup of patients. You can see here, this represents just over a quarter of cases. And when we look at the genes represented by this particular, we call this a risk signature, they're mainly associated with energy production in the mitochondrion. That's like the battery of the cell. And what basically, what this gene ACAP1 does is it switches on the recharging process. So after you exercise, the cell depletes its energy reserves, and this is the gene that switches those back on. So you would expect that patients who have defects in this particular system would be the ones showing up in the clinic with that profound fatigue, that post-exertional malaise. And actually, that's exactly what we do. So we use the phenotype and the survey data of patients to validate that the genetic hypothesis that we have is true. And we can differentiate this from the group next to them in blue here. These patients have defects in a neurotransmitter precursor transporter, so something that leads a gene that is involved in the production of particularly serotonin. And as opposed to this group of patients, this group you would expect, and they do, present with cognitive impairment, so that classic brain fog phenotype. But they also have enhanced levels of mood disorders. And when you go into the metabolomics, when you look at their blood and analyze the substances in it, there's an increased level of neurotransmitter precursors like phenylalanine. So we've gone from a really a hypothesis-free genetic analysis into clustering those genetic signals by the patients in which they co-occur, and then using the survey and the clinical presentation data to validate the fact that the hypothesis-free analysis has actually found things that are useful in a clinical setting. So that's the basis of what we do. One of the other genes that showed up here is clock. And clock is the absolutely classic regulator of circadian rhythm in a cell, the daily or diurnal changes in emphasis of energy production, sleep, the immune system. It's the master driver, the clockwork of the regulation of our cellular function. It's really interesting to see it showing up here because it also shows up in long COVID. This is one of the genes that maps between the two diseases. So a final thing just on our ME/CFS, one of the key things that is interesting here for the future is that these signatures turn out to be a lot more predictive of disease than we had any right to expect. Just rather like Heath was saying, these diseases are very difficult to study. In fact, there's never been a reproducible genetic association prior to this study in any ME/CFS population. But what we were able to demonstrate is that simply by counting the number of times one of those risk signatures showed up in an ME/CFS patient versus a control, we were able to get really rather stunning odds ratios for the predictivity of these. And obviously I wouldn't be showing this to you if that data didn't replicate across other disjoint patient populations. So we're investigating this, not least because it lends itself to a very simple genotypic test where from blood or saliva for a patient, very quickly, very cheaply, non-invasively, well, maybe a finger prick, we can actually validate not only that a patient likely has ME/CFS or long COVID or fibromyalgia or some other diagnosis that could be confused, but also the mechanism that is defective in that individual patient. And that opens a whole bunch of different doors. But I'm on to talk about long COVID and the results of the Sano study. So this is some of the data that we started with. So these are the samples collected through the different variant forms of the disease as the disease progressed through the population. We were using data that was looking at both the acute symptoms and also the long COVID symptoms, and particularly looking at some of the individual presentations of the disease, and also correlating those with self-reported comorbidities. We defined two different cohorts, one who had very severe disease, so they were not, they had the biggest change in health as a result of having long COVID, and they were not recovering. And we compared that against a group of patients whose symptoms seemed to be mainly about fatigue. So post-exertional malaise, breathlessness, inability to sustain physical activity. And we went in analyzing these two patient populations to understand where they were similar and where they were different. And through that process, we also demonstrated, I've got another line on the graph up here, we also went in and demonstrated that perhaps the incidence of long COVID was slightly declining. The other thing to note about this, other than the variants, is, of course, vaccination started to kick in just about the time that Alpha was becoming the dominant variant in the UK population. So we haven't been able to totally deconvolute those effects, but nonetheless, it's kind of interesting for follow-up studies to look at that. We were able, in the analysis of Sano's gold population, to identify 73 highly associated genes. 14 of those had been differentially expressed in other studies where you take cells and you look at which genes are upregulated or downregulated in those tissues. And that's a really interesting and useful confirmation that we might be in the right space biologically. 9 of the genes that we found in long COVID were also overlapping with ME/CFS, which is kind of interesting, and I'll come back to that. And then when we started to look at the differences or similarities between severe and fatigue dominant forms, we found that the 2 forms of the disease shared many of those genes. So 53 out of the 73 were replicated across the 2 different cohorts. Nonetheless, there were a series of genes that were unique to the severe form and some that were unique to the fatigue dominant form. And you can see that these represent or are related to very different types of processes, cellular processes in the cell, which may explain, why the virus's effects are so heterogeneous. One of the challenges that we have with the disease is it affects multiple organs. And, there are ACE receptors across all kinds of cells in the human body. So the virus gets everywhere. There may be pools of virus kicking around in the immune system, which lead to subsequent triggers of exacerbations. And what we can start to do is to tease apart the effect of those on particular tissues and particular presentations of the patients. So the genetic variants, this is all described in a lot of detail in the papers, so I'm not going to go into a lot of detail here, but I am going to highlight again that we saw the insulin receptor and the clock gene, these 2 major driver mechanisms. Insulin receptor drives energy production. Clock, as I said, is this master this sort of driver of processes in the cell. We saw those mainly coming out in the fatigue cohort. That's not surprising because that's the overlap with the ME/CFS side of things. So there's a lot of different approaches that we can potentially go at. Where I wanted to very quickly, before I hand back, talk about some really exciting work that we've done is starting to think about actively protected biology. So one of the big questions is, why do some patients get the disease and others don't? So this is some work that we did using those risk scores, those predictive diagnostic tools. Having built out a risk score, this is in ME/CFS, but we're doing the same thing in long COVID. Having built out a risk score, we're now able to go to a general population who do not have the disease, who do not have any of the physical symptoms, and basically score them. So take every single one through and say, do you have a high lifetime risk of getting this disease or a low lifetime risk of getting disease? What that allows us to do is to find those patients who have the highest possible risk scores. They've had all of the exposures, they've got all the genetic risk factors, but they don't get the disease. So then the question is why? And what we can do now is compare that cohort against the patient cohort to eliminate a lot of the disease-related biological signatures. and we find a series of actively protective genes and targets. And this is incredibly exciting. It opens the door for a new generation of prophylactic drugs. It opens the door for precision therapeutic DNA vaccines, which can be immunomodulating, or gene therapies, which can produce proteins that are actually going to protect patients against the disease. So we're really excited about this, not least because we've actually found some of these. So this again is an ME/CFS. You're looking at two different patient populations, independent of each other. We've run that same analysis and we found the same gene associated in both cases. And this is to do with the catabolism, the breakdown of a neurotransmitter precursor called tyrosine, which is in the dopamine system. And we know this has an impact on disease biology. So this is the very start of us using the results that we've generated through these studies to go into the future. But I'll talk more about that perhaps in the next section when we think about where this kind of research might take us.
Thank you very much, Steve. This was really interesting. And I think particularly the different phenotypes matching different genetic profiles and what that might mean in terms of treating those participants, these patients. We can imagine that someone affected at the neurotransmitter level likely will not respond in the same way that someone who has mitochondria, like disrupting processes at play. So it's really interesting. I'll share my screen again, and if we can go back into the presentation. So to your point, we're now going to talk a little bit about the impact of these findings on pharma companies and on patient and what that might mean, because I'm sure a lot of people listening to us today, depending on their level of understanding of genetics and scientific understanding, have the same question. Where does that lead us? And so we can talk a little bit about that, but from a pharmaceutical perspective, my understanding from your presentation, Steve, is that identify exactly what the genetic factor at play might be, and also how that combines with other aspects, ultimately pave the way for identifying potential drug targets or potential treatments, or identifying, to your point, people who might be at higher risk of developing long COVID or severe COVID, and potentially get enrolled into clinical trial that look at preventative treatments. Could you talk to us a little bit more about that?
Yeah, absolutely. We're actively engaged in all of those things that you just described. So the diagnostic piece is incredibly important here. It's really critical to be able to accurately identify patients who have a particular mechanism, a particular series of genetic defects underpinning their form of the disease, because that will tell you basically not only how they are likely to present in the clinic, but as you said, which drug is most likely to be effective for them. So we are engaged in a follow-up project to this, a project called LACOMI, with the Decode ME and Action for ME teams. It's not only in ME, it's also in long COVID. And it's really trying to use those insights that we've generated to do two things. The first one is to clinically validate that the diagnostics are useful in a clinical setting. And that's to do, again, two things. Number one, to identify that they have long COVID as opposed to ME/CFS or fibromyalgia or even MS. And that's by doing a single test where we can evaluate the mutations that that patient has and compare them to genetic profiles that are associated with each of those diseases. And so if they light up for long COVID, you have a differential triage tool that can be used very, very quickly, non-invasively in the clinic, and can guide a clinician to the right diagnosis. The next question is, what do you do about that? And this, in so you mentioned both novel drug targets and also repurposing. So novel drug targets is our bread and butter with biopharma companies, and understanding mechanisms that you didn't know were previously involved in disease is a really useful way of creating innovation and finding different opportunities to treat different aspects of the disease. What we're also doing, and what is potentially a lot faster to bring to market, is drug repurposing. And this basically means, if we identify that a particular gene is involved in a clinically relevant patient subgroup, that group I showed you in ME/CFS, a quarter of ME/CFS patients, that the gene may have actually been studied in the context of other diseases. And we've analyzed 50 different complex chronic diseases. So we know where that gene is relevant, and if it's been studied, where there is candidate drugs or even drugs on market, drugs that may have come off patent, that could be used to modulate that target in the right direction. And that offers a really evidence-led way of suggesting that that this drug should be used in a clinical trial. So if you put those two things together, the final thing that we're doing, we're now partnered with a really interesting clinical research institute studying complex chronic diseases. And we have the ambition this year to recruit 4,000 patients to actually do the clinical validation of that diagnostic, but then also within that to select cohorts of patients who have specific mechanistic defects, where that's a high prevalence in the population, where there are safe and well-tolerated medicines that could be used, and where the readout for elimination or amelioration of their symptoms will be very quick. And we're expecting to start running a number of those studies this year.
That's really exciting. And you can see just how much more fast-tracked, the potential access to treatment for patients might be in this case, particularly with drug repurposing. You would have imagined these drugs would have passed safety data already and would have been in the public for a number of years. And so there is the really exciting possibility that they might become available to the public a lot sooner. I think the last aspect we haven't touched upon yet is what does this mean in terms of the future? Because this is a really exciting use case for long COVID. But as you mentioned in your presentation as well, we're starting to see overlaps in terms of genes with ME/CFS. And I imagine that this approach can be replicated across several condition areas, particularly suitable for complex chronic disease. So it's a really interesting model here to accelerate the transition to precision medicine and make drugs available to a large number of people sooner. So I'd love for you to talk to us a little bit more about what's your perspective on that, on what that means for the future of medicine.
Well, I'm really excited. I mean, this is right at the forefront of bringing precision medicine approaches into complex chronic diseases. We do a lot. We've done 50 different diseases, as I said. We've done a lot in precision neuroscience, so neurodegenerative and psychiatric, other CNS conditions, women's health. So we've done this level of stratification in endometriosis as well, and again compared against the best meta-GWAS methods that are out there. In things like ALS, where there's just been this absolute paucity of treatments because it's such a heterogeneous disease, we have multiple targets partnered now with our collaborators. But there's actually some other things that you can do with this, and one of the realities, if you go into psychiatric disease, for example, one of the challenges is it takes years to get patients on medications that are actually going to work for them. So being able to use this to guide that selection of therapy and even combinations of therapies because of the particular makeup of an individual and the medicines that they're likely to respond to, I think that opens up a huge potential impact for the healthcare system. But in the long term, I think your second point here is for the future is really where we're going. This is all about prediction and prevention of disease. And I think that is the only way in which healthcare is going to become affordable. And if I give you one final example, we've been able to build a model for the prediction of complications of type 2 diabetes. So being able to predict 5 years before onset of symptoms who's going to progress to renal failure and the need for dialysis. Being able to predict that means you can deploy monitoring, lifestyle changes, even therapeutic interventions if you need. But you may prevent people entirely from encountering that life-limiting and incredibly expensive symptom. And that's a fabulous way of, using these type of tools to improve outcomes for patients whilst also reducing the cost of care. And we're absolutely going to need to do that in the future.
Yes, and I can really see how that might impact patients and also their healthcare providers in a really meaningful way. To your point, we were talking about ALS earlier. We've considered disease in the past as, one list of symptom and, a diagnosis. But what we're finding is that there are different angles at which at which we can come at it. And so people with different phenotype and different genotype might actually have quite a different progression or course of the disease. I think for healthcare providers, just being able to explain to a patient in particular what might happen for them and removing some of this uncertainty can be hugely powerful, in particular in diseases like ALS. But I think also people tend to perceive disease as a 0 to 1 event. Again, this is not quite a chronic, like if you think about lung cancer, you tend to have smokers thinking, I'll either have it or I won't have it. I know people who smoke who don't develop lung cancers. And actually, what's really interesting here is that it's just showing the continuum and the complexity of how disease take place. And I think, for someone to be told, you have higher risk of diabetes because your mother or your father had diabetes is very different than you have a high risk not only of developing type 2 diabetes, but also going into renal failure because of your own genetics. The message here is a lot more powerful in terms of how valuable lifestyle changes might be. So I find that really fascinating. Sorry, please go ahead.
I totally agree with you. And I think that informing the patient and getting them to play a role in the management of their health is going to be a really important consequence of this type of research across different diseases.
Yeah, if I may, I just wanted to jump in and say that I think that's particularly important in things like ME/CFS, where there are no biomarkers, and I think we've all heard people say, well, I'm tired all the time, I still get up and go to work. So I think it's incredibly powerful just to have a genetic diagnosis to show that there really is a biological basis to what's happening here.
Yeah, it really changed the way we understand these conditions. We've gotten a lot of really interesting questions from the chat. So I'm going to put some out there for you both to answer. Steve, there was one for you in particular. Your paper mentions potential cross-disease repurposing candidates for treatment of long COVID and other conditions. Could you elaborate on whether your biomarkers could be used to inform development of drug targets for this candidate? We touched a little bit upon that, obviously. How do you perceive the next steps once you have identified a potential target? How do you bring these insights into the real world?
So what we're planning to do later this year is to use the diagnostic tools that I described as a way of identifying patients who have a specific mechanistic problem, so that is amenable to treatment by one of those drug repurposing candidates. What we can do then is recruit a small cohort of patients, maybe N of 30, N of 50, in an investigator-led study, and give them that drug to demonstrate that they benefit or they don't benefit in a clinically meaningful way from that treatment. If all of those things hold true, then there is a possibility of very rapidly getting approval for that medicine, because it's already, as you said earlier, we will choose these where they're safe and well tolerated and have passed phase 3, so they can be used for this application very, very quickly. And I think that is something that we can do not only in the context of long COVID and ME/CFS, but also we're trying to do in ALS and in endometriosis, as well as other diseases.
Yes, there are so many use cases for this approach. It's really interesting. We have another question that we haven't really quite covered in this presentation that I think is quite interesting as well. It was around how genes related to the immune system response to COVID-19 vaccines might play a role into this. I mean, you mentioned that obviously the change in prevalence in the virus changed at the same time as we introduced vaccine, but it would be potentially quite interesting to understand if some people genetically respond better to the vaccine, get a higher level of protection than others. What's your thought about that?
That would be absolutely fascinating, and particularly I think the strength and longevity of the immune response would be really interesting to compare and contrast, for example, between the mRNA vaccines and the ChAdOx and others. The challenge is that these effects read out across multiple organs in the body, and they affect different people to different extents in different organs. So it is just an incredibly complex picture of the underlying biology. But yeah, the interaction of the host genome, the immune system of that individual, which is, has its own variability, and then the triggers that the pathogen is capable of eliciting are really there's a lot of work to be done there.
Yes, I imagine. I mean, I can see the power of this in all condition, but I can imagine looking at endometriosis or ALS is slightly different, because here we have to consider the interplay between the host and the pathogen as well. And again, that's something we didn't touch about in the presentation, but you did do a bit of analysis segregating the different variants. And so there is also almost a question as to whether individuals with a specific genetic background might be more susceptible to long COVID with one variant versus the other. It might be then that one variant, for example, is more successful than its predecessor at targeting specific pathways.
Absolutely. Unfortunately, in the study that we did, we just didn't have enough sample numbers to convincingly or statistically significantly answer that particular question. We did try to look, but unfortunately, we just didn't have enough data.
Thank you. And I think we have time for maybe one more questions. We touched a little bit upon that during the presentation, and maybe I could get both your points of view on that one. Is there a genetic predisposition to post-viral conditions beyond long COVID? And what controversies are there in this area? So I think that's quite an interesting topic, because you clearly showed that there was overlap, in some genetic factors in long COVID versus ME/CFS. And I think it's hugely exciting to see that all of the resources that have gone into studying COVID could now potentially pave the way for, game-changing research in ME and CFS. But how do you sort of perceive the differences between the two, and how do you entangle this from a research perspective?
I could give a personal anecdote if I might, which is that I was telling people for several months that I thought I had long COVID because I had like acute symptoms and then just took months to recover my energy levels. But then I eventually took an antibody test and it was negative. So it turns out I'd never had a COVID infection and it was some other post-viral syndrome. So, I mean, I do think this has been going on for a long time and just hasn't been recognized because of the acute severity of this particular pandemic.
That's really interesting. I wonder whether because COVID was introduced, on a mass scale, like we all get ill throughout our lifetime, but I guess the timing at which, any given person develops the flu or other viruses tend to be scattered across a different timeline. So COVID created a little bit of this mass effect where suddenly everybody started paying attention to a process that, might have been happening for quite a long time in response to viral infection. Steve, I'd love to hear your thoughts on that.
Yeah, I mean, it's a complex and evolving picture, and I think it's going to take a lot more disentangling of the effects of the virus and the post-infection sequelae, and the impact that they have through subsequent viral infections. It seems that, other viral infections, whether they're COVID or not, can actually trigger some exacerbations of fatigue, for example. But there are some other issues that we need to pay attention to. So there is some evidence that for patients, particularly who have multiple COVID infections, there is about a doubling of the cardiovascular risk for those patients. We also see a lot of overlap with genes that are associated with neurodegenerative diseases in our studies. So Alzheimer's, ALS, Parkinson's. And anecdotally, there was a significant uptick of Parkinsonism several years after the Spanish flu, quite widely across the world. So I think from a public health perspective as well, it's going to be necessary to keep an observational epidemiological lens on this, and then start, if we do see populations arising with specific issues, again, to go back and see whether there are genetic or other stratifications that could be done to identify patients who are most at risk, and perhaps those are populations who need more support in terms of therapy or other disease prevention measures.
Thank you. I think we've come to the end of our webinar. If there are more questions, please do send them via email or through the Sano website. We'll be very happy to answer them as well after the webinar. The recording of this webinar, as well as the resources, are going to be made available both on the Sano Genetics website and PrecisionLife, and on social media as well. So please don't hesitate to go and look for these resources if you want to dig a little further or go back on the slide. Steve, Heath, I want to extend a huge thank you to you both. I've learned a lot today. It's been a really interesting presentation and hugely exciting to see just how close we may be to unlocking access to treatments for a range of conditions that have been affecting people chronically for years, and where we felt like maybe we had reached the limit of what medicine could do. This approach is obviously opening doors to a lot more possibilities. And I think that's a really good note to end this presentation on, because I would say watch that space. There is a lot to come. Thank you very much for your time. And yes, please feel free to send questions our ways.